Hi, everyone. My name is Brandon Cuevas, and I'm here with Benjamin Greenfield
Howdy.
and Benjamin Kepes.
Hello, everyone.
And we are students at Columbia University, part of a club called the Synthetic Biology Initiative, whose mission is to foster science learning and advance the interest and public understanding of synthetic biology. So, essentially, what we plan on doing and what we seek to do is to spread knowledge of this rapidly evolving field. This relatively new field, synthetic biology, has only been around for 10 years, and it's not as well developed as other disciplines, which is why we plan to launch this lesson plan and video series to educate those who are interested in learning more about synthetic biology. So, what exactly is the point of this series? This series will mainly focus on molecular cloning, which is a major tool in biology research, especially in the last decade. Right? It includes things such as gene editing, manipulation of bacteria, basically genetic engineering, which is helpful in therapeutics and agriculture. It has a wide variety of uses, and the point of this series is to introduce you to the basics and cornerstone of what makes up genetic engineering and also what makes up synthetic biology, because synthetic biology is a large part of manipulating such biological systems in order to improve them and make them function in a way that we haven't even imagined before. So whether you are a beginner, intermediate, or expert, we seek to talk about many different areas of synthetic biology in different levels. So we hope to satisfy any of you guys, despite your experience with biology or even synthetic biology specifically. But without further ado, let's actually get into talking about what molecular cloning is.
So, what is molecular cloning? Molecular cloning is the way scientists alter bacterial DNA and create new recombinant organisms. We'll explain more of the specifics later, but an example of the use of molecular cloning is when scientists can make bacteria that produce something we want, like a protein, a drug, or a food product. Why is molecular cloning important? For one, it's an important technique that genetic engineers do. There are thousands and thousands of papers published in which the authors use molecular cloning, but it's so useful in many fields of science. It's also useful in medicine. For example, modified bacteria are used to produce human insulin for diabetics.
So there are many ways to engineer bacteria. In fact, every year scientists are coming up with more ways, more tools that we can use to alter the DNA of bacteria and achieve the results that we want. Every method for engineering bacteria has its advantages and disadvantages, and everyone has their own personal preferences as to which method to use. Some examples of these that we're going to be talking about in much more detail later are ligation. This was one of the first methods of making new bits of bacterial DNA that we wanted. Ligation involves the use of special enzymes called restriction enzymes, which cut DNA at very specific places, and then we can use the sticky ends in the DNA left by these enzymes to reseal the DNA fragments in the ways that we want. And it's all right if you don't know what any of this means yet. We'll talk about it more later. Another method that a lot of molecular biologists use nowadays is Gibson. Gibson is a really great, straightforward method for engineering bacteria that involves using overlapping strands of DNA to make new plasmids. And some other names that I'll drop here are Golden Gate Assembly, CPEC. These are all molecular cloning techniques, and we're going to talk about all of these in future videos. But the first thing we'll do before all of that is we'll give you a brief overview of the molecular biology involved in all this molecular cloning, and that will be lesson 2. So until then, this has been Ben Kepes, Brandon Cuevas, and Benji Greenfield. Thank you.